Welcome to the deep dive. We're here to really get into the details of complex research.
Making it accessible.
Exactly. And today we are tackling a really big one, a fundamental assumption in ecology and conservation all over the world. The idea that an organism's its external features should basically predict its job in the ecosystem, its function.
Yeah, form equals function. It's deeply embedded. We use it constantly when managing ecosystems, assessing biodiversity, assuming that things like body shape, mouth size, fin position tell us what an animal, especially a fish, does, what it eats.
And this isn't just, a theoretical nicety. It genuinely guides huge conservation strategies, global biodiversity reports. It's foundational.
Absolutely.
So what happens when you take that core belief and put it through the most rigorous global test ever done?
That's precisely what we're digging into today. There's this paper, Looks Can Be Deceiving. Morphological traits are weakly associated with the isotopic niches of freshwater fishes. The lead author is Julian D. Olden, working with a big international team.
Looks can be deceiving. I like that title.
It's appropriate. And this paper is a necessary reality check. Honestly, maybe a bit of a shock for anyone relying heavily on those big fish trait databases.
Okay, so let's nail down the mission here. The goal of this research and our deep dive is to test on a global scale whether the physical appearance, the morphology of freshwater fish actually gives us reliable clues about what they really eat, their diet in the wild.
It's super critical because these functional traits, particularly the easy-to-measure morphological ones, they're the currency we use. They're compiled in huge databases like FishMorph.
Right.
And scientists use them all the time as a stand-in, a proxy for trophic ecology, what fish eat when deciding conservation priorities or measuring functional diversity.
Because it's easier than checking stomach contents for thousands of fish.
Exactly. But the problem, or the gap, was that we didn't have a really solid global scale check to see if this shortcut actually works until this study.
Okay, so performing this massive check required, equally massive data sets, right? They needed the looks data and the actual diet data. What did that data stack look like?
Oh, the scale is just incredible. They pulled together data on 1,282 fish species.
Wow.
From over 800 distinct fish communities. And get this, spanning 65 countries, with data collected over 3 decades, from 1992 right up to 2023.
That is a truly global effort, immense scope.
It really is. And for the morphology, the looks part, they focused on 10 specific external traits, things we think are relevant to feeding.
Like
Overall body length, the general body shape, where the mouth is positioned, eye size, fin placement. The kinds of things you can often measure pretty easily, even from a 2D photograph.
Which is why they're so popular in those databases, right? Easy data collection.
Precisely. Cheap, fast, scalable.
Okay, so that's the morphology side. What about measuring the actual diet? You said not stomach contents. So how did they figure out the trophic niche?
They used a really sophisticated chemical approach. Stable isotope ratios. Specifically, delta C13 and delta N15 dollars. They got this data from another huge database called IsoFresh.
Okay, isotopes. Quick refresher. What do those specific signatures, C13 and N15, tell us about what a fish has been eating?
Right. So think of stable isotopes as a kind of long-term chemical fingerprint of an animal's diet. It's not just the last meal, like stomach contents. It integrates diet over time.
Okay.
The delta dollars, the carbon isotope ratio, tells us about the source of the carbon, basically where the food came from. Did the fish mainly eat things living in the water column, pelagic stuff, or did it feed more on things down on the bottom, benthic resources?
Got it. Carbon source.
Yes.
And the nitrogen, delta dollars.
That's the really cool one for trophic ecology. Delta Reshadiler reflects the consumer's trophic position. It tells you where that fish sits on the food chain. Is it eating plants? Is it eating herbivores? Is it a top predator eating other fish? The higher the delta dollars, generally, the higher up the food chain it feeds.
Okay, this is where it gets really interesting then. We have this massive global data set comparing the easy-to-measure looks, the morphology against this rigorous chemical measure of the actual long-term diet, the isotopes, and the results. Well, they hit that core assumption pretty hard, didn't they?
They really did.
What did the main global analysis, the multiple regression on distance matrices, the MRM, what did that show about the link between looks and diet?
The link was weak.
Right.
Just consistently weak across the board, averaging across all 811 of those communities. Similarities in how species looked, their morphology, explained only about 10% of the variation in their diets as measured by the isotopes.
10%.
10% on average. Think about that. It means 90% of the difference in what fish species we're eating had basically nothing to do with the external physical traits we usually measure.
That's quite low.
It's incredibly low, considering how foundational this assumption is. And it gets even starker statistically significant associations between looks and diet. They only found those in just over 1 in 20 communities, about 5.9%.
Wow, okay. 10% average explanatory power is.
Yeah, that feels pretty devastating for relying on morphology alone. But they also used other models, right? The MGLMs, multivariate generalized linear models. What did those add?
Right, those models tried to explain the variation in the isotope values using both the morphology traits and the site where the fish were found. And they did explain a decent amount of the variation overall, about 53% for the carbon source and 48% for the trophic position.
Okay, so the models worked reasonably well.
Well, and this is the crucial point. The morphological traits themselves contributed almost nothing to that explanation. They barely registered.
So where did the explanatory power come from, if not the traits?
This is the absolute key finding.
Yeah.
The vast majority of that explained variation. An incredible 97% for the carbon isotopes and 77% for the nitrogen isotopes was explained by the site identity, just knowing which lake or river system the fish came from.
So the location, the specific environment. Exactly. Where the fish lived mattered vastly more in predicting its diet than its physical structure did. It suggests the local conditions, what prey is actually available, the water temperature, maybe predation risk, habitat structure. These factors are so powerful they essentially overwhelm, or at least massively overshadow, that expected link between the fish's form and its function.
The location dictates the menu far more than the fish's build does.
That's a great way to put it.
Okay, but hold on. Did any of the 10 morphological traits they measured show any predictive power at all? Was anything linked to diet, even weakly?
Minimally.
Yeah.
Really minimally. There were only 2 traits that showed any significant relative importance, and only for predicting the trophic position, the delta dollars.
Which ones?
Oral gape length, basically how wide the mouth opens, which relates to the maximum size of prey a fish can swallow.
Makes sense.
And maximum body length. Bigger fish eating higher up the food chain, potentially.
Yeah.
But combined, these 2 traits explained only about 12% or so of the variation in trophic position.
So, still pretty small.
Very small. It gives a tiny bit of support to the basic idea that, yes, body size and mouth size put some constraints on prey size, but they are absolutely not reliable predictors of the overall diet across the board.
And I remember seeing something about different feeding guilds. Did the connection differ between, say, herbivores and carnivores?
Yes, absolutely. That was another interesting wrinkle. For herbivores, the plant and algae eaters, the link between their morphology and their diet, specifically their trophic position, delta dollar, was significantly weaker than it was for omnivores or carnivores.
Huh. So for plant eaters, their external looks tell us even less about their diet.
Seems that way. Perhaps their external form is driven more by other factors, or their diet is even more context-dependent.
Okay, that covers the broad species-level picture. But the researchers didn't stop there, did they? They also looked within species. Intraspecific variability.
Right. They drilled down to see if maybe, just maybe, the physical differences between individual fish of the same species could explain why those individuals have slightly different diets. They did detailed case studies on 5 species, including familiar ones like largemouth bass and European minnow.
And what did that finer-grained analysis show? Did individual looks predict individual diets better?
Sadly, no. The results at the individual level pretty much mirrored the big picture. On average, across those 5 species, individual differences in morphology explained only about 4% of the variation in their isotope signatures.
4%, even lower.
Yeah. The physical build of one specific base, for instance, told you very little about what that particular base was eating compared to another base in the same place. The range was basically 0 up to maybe 16% at the very maximum.
Well, even zooming right in, a fish's specific body shape or mouth size isn't a reliable guide to its personal menu.
Mostly correct. There was one interesting exception, though.
Which was?
The largemouth bass. That species actually showed the strongest association between individual morphology and diet within the case studies.
Why bass?
Well, the researchers suggest it's likely because largemouth bass undergo a really strong and pretty predictable change in diet as they grow. It's called an ontogenetic shift. Basically, as they get bigger, they switch much more heavily to eating other fish piscivory.
So size really does dictate diet in that specific case because bigger bass fundamentally eat different things than smaller bass.
Exactly. In that scenario, body length becomes a much more direct predictor of diet because of that strong, size-driven dietary switch. But that seems to be more the exception than the rule.
Okay, that exception is really interesting because it helps highlight the general weakness, doesn't it? This lack of connection seems to tie into a concept ecologists talk about, Leem's paradox.
Precisely. Leem's paradox is this idea that an animal might possess highly specialized physical structures, the potential to eat a very specific type of food.
Like teeth perfect for crushing snails.
Right. But if other food sources are really abundant, or maybe competition for that specialized food is high, or the specialized food is scarce, that animal might just act like a generalist. It eats what's easy and available.
So its realized diet, what it actually eats day to day, isn't dictated by its specialized tools, but by the ecological context, opportunity.
That's the core of it. The potential form doesn't always match the reality function in a given environment. The pumpkin seed sunfish is the classic example you mentioned. They have those specialized pharyngeal jaws great for crushing snails.
Yep.
But if they live in a lake where snails are rare, or maybe where bluegills are abundant and easier to catch, they'll shift their diet completely to soft-bodied insects or other invertebrates. Their specialized morphology is still there, but it's decoupled from their actual diet in that context.
And that brings us back to that staggering finding from the MGLMs that 97% of the carbon isotope variation and 77% of the nitrogen was explained by site identity. It screams context dependency, doesn't it?
It really does. It strongly suggests that the local environment, the prey available, the predators present, the water conditions, is just so influential that it masks or overrides whatever signal the fish's morphology might otherwise provide about its diet. The form-function relationship gets blurred by reality.
Okay, so if we zoom out again and think about the bigger picture, what are the main implications here? What's the critical takeaway from this huge study?
I think the biggest takeaway is a really strong note of caution. It cautions us against blindly accepting that unspoken assumption that easily measured external morphological traits are reliable indicators of trophic ecology, especially when we're doing broad-scale ecological studies or making big conservation plans based on functional diversity metrics derived from these traits.
We might be oversimplifying things by relying on these convenient proxies.
We might be. The paper basically says, hold on, let's be more critical about this. We need to be really honest about the limitations when we label traits as functional, just because they're easy to measure and readily available in databases.
And speaking of limitations, we should also acknowledge the limits of the method they use to test the assumption, right? Stable isotope analysis isn't perfect either.
No method is perfect, and the authors are upfront about that. Stable isotope analysis, or SIA, is powerful because it gives that time-integrated picture of diet, which is great. But it has its own uncertainties. Things like differences in lipid content fat levels between species can affect the carbon isotope values, for example. And the baseline isotopic signatures of the food web itself can vary from place to place, which needs careful correction.
They did correct for baseline variation, though.
Oh yes, they performed thorough standardizations, which is crucial. But those inherent uncertainties in SIA itself mean we always have to interpret the results, even these results, with a degree of scientific caution.
Okay, so if external morphology alone is a weak predictor of diet, where do we go from here? Should fish ecologists just, I don't know, give up on using morphology?
Not give up, but perhaps broaden the focus significantly. The paper makes a strong case for moving beyond just these easily measured external traits.
Towards what?
Towards incorporating internal anatomical traits, structures that are much more directly involved in the actual mechanics of catching, handling, and digesting prey.
Like what kind of internal structures?
Things like the structure of the gill rakers, which filter food particles, the detailed mechanics of the jaw, the type and arrangement of teeth dentition, even the length of the intestinal tract, which relates to digestive strategy. These internal features are likely far more tightly linked to feeding ecology than, say, how far apart two fins are.
That makes intuitive sense, but measuring internal structures on thousands of fish sounds challenging. Much harder than taking a photo.
Historically, yes, but the technology is catching up. This is where it gets exciting. The paper specifically mentions the promise of new tech, particularly microcomputed tomography, micro-CT scanning.
Like tiny CCAT scans for fish.
Essentially, yes. These advanced scanning techniques allow us to non-destructively visualize and measure these complex internal structures in great detail, and potentially at a much larger scale than was ever possible with dissection.
So we could potentially build databases of internal functional traits.
That's the hope. It could give us a much more robust and biologically meaningful picture of fish trophic ecology. The future likely lies in combining multiple lines of evidence, maybe improved morphological data, including internal, large-scale diet databases like stomach contents where available, and stable isotope databases to get a more complete understanding.
Okay, so let's try to summarize the core message then. What this deep dive into the Olden paper tells us is that the visual appearance of a freshwater fish, its external shape and features, while maybe useful for understanding how it moves or what habitat it prefers, is generally a pretty weak proxy for its actual realized trophic niche. What it really eats in its specific, complex, highly context-dependent environment seems to be driven much more by that environment itself.
Well put. The location, the context, seems to dictate the menu much more strongly than the fish's external toolkit does, in most cases.
Which leaves us with a really provocative final thought, something the paper raises directly.
Yes, it's a fundamental question for the field. If these commonly used external morphological traits are in fact so weakly associated with the trophic function they're supposed to represent,
Right.
then should we even be calling them functional traits in this context? Or are they perhaps just traits, traits that we as ecologists have functionalized mostly out of convenience because the data is available and easy to gather?
That distinction between a trait being truly functional for predicting diet versus just being available, that's huge.
It really is. It has massive implications for how we interpret functional diversity, how we model food webs, and ultimately how we make conservation decisions based on these large-scale data sets. It's something everyone using these trait databases needs to think about very carefully.
A fascinating and potentially field-shifting question to end on. Is it function? Or is it convenience dictating our labels? We encourage you, our listeners, to think about that, maybe consider other areas in science or just in life where we might assume form dictates function, but perhaps the reality is a lot more complicated.